After studying this module, you shall be able to know about the significance of neurotoxic poisons, origin and classification of neurotoxic poisons, mechanism, forensic importance and analysis of neurotoxic poisons. Most multicellular animals possesses a nervous system. In every illustration, the purpose of the nervous system is to receive information about the external and internal environment, assimilate the information and then direct a response appropriate to the stimulus. In addition to these elementary vital functions, the nervous systems of higher organisms are responsible for sensitivity, thinking and learning. All of the other organ systems of the body are subject to control by the nervous system. Therefore, impairment to this controlling system by toxicants can have comprehensive and even devastating effects. In vertebrates, there are two main components of the nervous system. Although these two systems are anatomically distinguishable, they are attached and they function interactively. The brain and spinal cord comprise the central nervous system and the nervous tissue, which is ganglia and peripheral nerves, outside the brain and spinal cord comprises the peripheral nervous system (PNS). Many substances alter the normal activity of the nervous system. Sometimes these effects are instantaneously and momentary, like the simulatory effect of coffee or a headache from the fresh paint. Other effects can be much more deceptive, like the movement disorders suffered by mine workers after years of chronic manganese intoxication. Many agents are harmless or even therapeutic at lower doses but become neurotoxic at higher levels. Neurotoxic poisons are the poisons which affect the central nervous system (CNS). Neurotoxicants and toxins have been comprehensively studied because of their toxic effects on humans and their effectiveness in the study of the nervous system. It is estimated that millions of people worldwide are exposed to these neurotoxicants every year. A static data emphasized by repetitive occurrence of neurological disease. Neurotoxicity is defined as an adverse change in the structure or function of the nervous system following exposure to a chemical, biological or physical agent. A significant theory in calculating neurotoxicity is that of the selective vulnerability of the nervous system to injuries, which is based on the structural complexity and cellular and molecular heterogeneity of the nervous system. Neurotoxicants can preferentially affect particular neuroanatomical regions, and with those regions the target can be a specific type of neuronal or glial cell. The most essential property of nervous tissue is its ability to transmit information. This is accomplished by the transmission of electrical or chemical signals. These processes are open to disruption by chemicals. Electrical signals in the neuron are transmitted by the movement of ions across the membrane through different ion selective channels. These channels can habitually act as the focus of the neurotoxicant action. Chemical signals are propagated by the presynaptic release of neurotransmitters that diffuse across intercellular spaces and interact with receptors on adjacent cells' membranes. These neurotransmitters, receptors, and related second messengers, anabolic and catabolic processes may also be the target for neurotoxicant action. At the molecular level, neurotoxicant may act on nervous tissues by interacting with a range of biochemical processes. The nervous system is also a common target of toxic metals, mainly organic metal compounds. For example, methyl mercury, because it is lipid soluble, readily crosses the blood brain barrier BBB and enters the nervous system. By contrast, inorganic mercury compounds, which are more water soluble, are less likely to enter the nervous system and are mainly nephrotoxicants. Similarly, organic lead compounds are mainly neurotoxicants, whereas the first site of inorganic lead is enzyme inhibition. Forensic issues. Although awareness of the toxicants' complete biochemical or molecular mechanism is the crucial objective of neurotoxicology, a comprehensive understanding of the toxicity also necessitates knowledge of the functional consequences of those changes, being the final output or manifestation of the nervous system functions includes motor, sensory, autonomic and cognitive capabilities. It is clear that neurotoxicants can stimulate a broad spectrum of biochemical, structural and functional abnormalities. This multiplicity must be taken into consideration in screening unidentified chemicals for the potential to produce neurotoxicity. Many different neurochemicals have been recognized and measured in tissues of the nervous system. These contains neurotransmitters, receptors, second messengers and metabolic and catabolic enzymes. The presence of a variation in a particular neurochemical parameter is not as such an indication of a toxic effect, since many neuroactive compounds can cause pharmacological changes. Many neurotoxicants function by inhibiting the GABA receptor, resulting in prolonged closure of the chloride channel and excess nerve excitation. Cyclodiene insecticide means dilidrin, the organochlorine insecticide, lindrine, and some pyrethroid insecticides all provoke acute neurotoxicity, at least in part, throughout this mechanism. Symptoms of GABA inhibition includes dizziness, headache, nausea, vomiting, fatigue, tremors, convulsions and death. Now, the classification of neurotoxic poisons. Neurotoxic poisons are categorized into three groups. First is the cerebral neurotoxic poison. In this, as resulted by the term, these poisons generally affect the cerebral region of the brain. Cerebral neurotoxic poisons can further be categorized into following classes. In this, the first is somniferous poisons. These drugs generally produce soporific effects. Somniferous poisons are one which produce numbness and sleep and are often used therapeutically. Example, opium derivatives and pethidine, etc. Now the second is inebriant poisons. Inebriant poisons are substances that induce intoxication or inebriation, that is, a state of excitement and necrosis. They are further divided into 5 subclasses. In this, the first is classical inebriants, alcohol, which means ethyl alcohol, methyl alcohol, and ethylene glycol. Second is sedative and hypnotic, in which chloral hydrate and barbiturates are there. Third is insecticides. In this, the organophosphorus compounds, organochlorines, carbamates. Fourth is the hydrocarbons. In this, the aromatic hydrocarbons like benzene, aliphatic hydrocarbons like petroleum products, halogenated hydrocarbons like carbon tetrachloride. And the fifth is the benzodiazepines. In this, the diazepam and flurazepam, etc. are present. Now, the third point is the deliriant poisons. Deliriants are substances that induce delirium, which means acute confusional state with disorientation, delusion and hallucination, and comprise mainly dhatura and cannabis. Other examples contain belladonna, hyoscyamus, cocaine, etc. Now, the spinal neurotoxic poisons. Spinal neurotoxic poisons are those poisons which act upon the spinal cord. Example contain Strychnos nux vomica, glencium and phygostigmine, etc. Now, the peripheral neurotoxic poisons. Peripheral neurotoxic poisons are those poisons which act especially up on the end plate of the motor nerves terminal. Example include hemlock, cocaine, conium, etc. Now, the miscellaneous neurotoxic poisons. In this, the first is stimulants. There are several drugs and chemicals that stimulate the central nervous system, among which the most important are the anesthetics. Anesthetics are used for the controlling of pain in many medical and operative procedures. Anesthetics may in themselves result in several adverse effects and occasionally death of the patient. In contrast to earlier years, anesthesia is related with low mortality nowadays and is estimated to be 1 in 10,000 with proper care in administration. There is evidence to suggest that operating room staffs may suffer from the results of occupational exposure to general inhalation anesthetics if safe measures are not taken. Anesthetic agents are not uncommonly employed for suicide, but homicides are exceptional. There are additional perils in every operation under general anesthesia. Local anesthetics are absorbed from mucosal surfaces as well as from parental sites. They are rapidly absorbed from the gastrointestinal tract with peak plasma levels occurring within 1 hour. Toxicity is due to exaggerated pharmacological activity, principally involving the cardiovascular and central nervous system. High drug levels result in depressed membrane function of all excitable tissues, ultimately resulting in membrane instability. Now, the anesthetic agents can be classified into two groups. General anesthetics. In this, the first is inhalational agents. Inhalational agents are the anesthetics which are given through the respiratory tract. For example, it includes nitrous oxide, halothane, enflurane, sevoflurane, and desflurane. Now, the intravenous agents. These anesthetics are injected intravenously to induce slumber. The examples include diazepam, ketamine, propofol. Now the local anesthetics. It contains cocaine, lignocaine, benzocaine, etc. falls under the category of local anesthetics. Now the psychiatric drugs. The main drugs used in psychiatry contains those specified for the treatment of anxiety, psychosis, depression and mania. Major depression is one of the affective disorders and is possibly the commonest mental illness worldwide. It is characterized by feelings of deep sadness and desolation, slowing of thought process, impairment concentration, persistent anxiety, agitation and self-depreciation. Migraine is treated pharmacologically using either an acute or prophylactic or combined medication program using one or more or several drugs available for the purpose. These drugs include analgesic like aspirin, antiemetics like diphenhydramine, combinational analgesics like aspirin, ergot alkaloids and sumatriptans. Pyrolactic treatment largely involves beta blockers, calcium channel inhibitors, serotonin and antagonists and antidepressants. Now the anticonvulsant drugs. Anticonvulsant drugs are which fight convulsions of both the epileptic and non-epileptic variety. They are further classified into the following classes, in which hydantoins, barbiturates, deoxybarbiturates, immunostimulants, succinamides, oxalobenzodiazepines and other anticoagulants are present. Now the psycholytic drugs. Psycholytic drugs, also known as hallucinogen, are substances that induce alterations in thought, perception and mood without causing major disturbance in the autonomic nervous system. Perceptual alterations can take the form of illusions or hallucinations. Few of the psychedelic drugs are main cause of death, but some may lead to traumatic deaths because of the abnormal behavior of the person who is under its influence. The hazards of hallucinogen use do not have as much to do with the acute toxicity as with the long-term psychological damage. The unavoidable consequence is violent crime manifesting as assaultive behavior, homicides and suicides. Several horrendous crimes have been committed by drug-crazed individuals acting out their inexpendable imaginations. Now, the mechanism of neurotoxic poisons. Neurotoxicants affect the nervous system in a number of different ways. Some neurotoxicants damage the distal portion of axons without much effect on the remainder of the cell, while others produce absolute cell death. Still others affect signaling processes in the nervous system without causing structural damage. This wide variety of neurotoxicant effects is due in part to the distinctive nature of the different types of neurons and glia. When assessing the potential neurological effects of a compound, effects may be on the central or peripheral nervous system or related to exposure that occurred during development. To understand the mechanism of action of individual neurotoxic compounds have originated with the identification of the cellular target. In the nervous system, this has most often been one of the four targets, which are neuron, the axon, myelinating cells, and the neurotransmitter system. As a consequence, neurotoxic compounds may be recognized which cause neuronopathies, angiopathies, or neurotransmitter associated toxicity. Now, the neuronopathies. Certain toxicants are specific for neurons or occasionally a particular group of neurons, resulting in their injury or when intoxication is severe enough, their death. Some of the exclusive features of the neuron include a high metabolic rate, a long cellular process that is supported by the cell body, and an excitable membrane that is rapidly depolarized and repolarized. Since many neurotoxic compounds act at the site of the cell body when massive loss of axons and myelin are discovered in the peripheral nervous system or the central nervous system, the first question arises whether the neuronal cell bodies themselves have been destroyed. Although a large number of compounds are known to produce toxic neuropathies, all of these neurotoxicants share certain features. Each toxic condition is the result of a cellular toxicant that has a preference for neurons, most likely due to one of the neurons' uncharacteristic exposure. The initial injury to neurons is followed by apoptosis or necrosis, leading to perceptual loss of the neuron. These chemicals tend to diffuse in their action, although they may slow some selectivity in the degree of injury. Now, the axonopathies. The neurotoxic disorders termed axonopathies are those in which the primary site of toxicity is the axon itself. Axonopathies can be considered to result from a chemical transaction of the axon. The number of axonal toxicants is outsized and increasing in the number. In this type of injury, the primary site of toxicity is the axon and it produces an effect that is the chemical equivalent of the axonal transaction. This generally leads to the degeneration of the axon distal to the transaction point, followed by the secondary degeneration of the myelin sheath. The cell body itself may be spread. Axonopathies have been further classified as central, peripheral, distal axonopathy to indicate those injury primarily involving the distal ends of long axons. Now, the myelinopathies. Myelin provides electrical insulation of neuronal processes and its absence leads to the slowing of and or abnormal transmission of impulses between adjacent purposes purported as apophatic transmission. Toxicants exist that result in the separation of the myelin lamella termed intramyelinic edema and in the selective loss of myelin termed as demyelination. The compounds such as ethidium bromide, which is ETBR, etc. leads to myelinopathy. Some of these compounds have created complications in humans and many have been used as a tool to explore the process of myelination of the nervous system and process of remyelinating of following toxic disruption of myelin. Now, the neurotransmission associated neurotoxicity. A wide-ranging diversity of naturally occurring toxin as well as synthetic chemicals alters specific mechanisms of intercellular communication. Although neurotransmitters associated actions may be well understood for some agents, the specificity of the mechanisms should not be assumed. Amphetamine, atropine, cocaine, domoic acid, etc. are related to neurotransmission associated neurotoxicity. Organophosphates and carbamide pesticide produce their insecticidal action by inhibiting acetylcholinesterase, the catalytic enzyme that ends the post-synaptic action of acetylcholine. The constitutional cholinergic overstimulation produces signals of acute toxicity ranging from flu-like symptoms to gastrointestinal distress, ataxia, twitching, convulsions, coma, and death. These effects are not as well correlated with acetylcholinesterase inhibition as might be expected for all such pesticides, leading to suggestions of additional mechanism of actions that have since been verified in animals and in vitro studies. These include direct actions on pre and post synaptic cholinergic receptors and different reuptake of choline. Such actions serve to modulate the downstream influence of cholinergic overstimulation. Thus, multiple neurotransmitters targets may be more common than was once expected. Now, this summary. Neurotoxicology is the study of toxic substances and their possible effects on functions and activities of nervous systems. Thus, it is the study of capability of an agent to adversely affect the structural or functional integrity of the nervous system. The nervous system is unique in structure and remarkably complex, intricately sensitive, nevertheless depth of incredible adaptability. Because of these attributes, the neurotoxic potential of many agents, to say little of their core mechanisms, remain unknown. Many creatures of the animal kingdoms, example snake, contains neurotoxic venoms. Cobra, coral snakes, etc. possesses extremely toxic neurotoxin which can paralyze and cause death of the victim in a short span of time. Besides, some insects also contain neurotoxic poisons in their stings. Many chemical agents and metals such as chlorinated hydrocarbons, bismuth, mercury, selenium, etc. have neurotoxic properties. Some Clostridium species produce natural neurotoxins which are deadly in nature. Apart from that, mycotoxins produced by the fungi also tend to produce brevetoxins produced by the dinoflagellate. Gymnodinium breve is considered to be the most potential neurotoxin known.